Good afternoon, everybody. This is Luke D, and this is Honors by Two Mechanisms of Cancer. And this is a quick introduction, no, not introduction, but quick explanation of genetics. First of all, this is the genetics problem set I want you to complete for homework. It is 3 problems. I want you to work on it independently. Um, these shouldn't be too hard for you. Do not try to use Punnett squares here because they will probably mess you up. If you just use straight probabilities, this should be a pretty easy assignment. Anyway, this will be in the Dropbox for you, so you can access that whenever. So what I thought I would do is show you the typical genetics problems that I seem to have such a problem with. Now, I want you to remember that if most things work like this, then diseases like diabetes and obesity and addiction and Alzheimer's and cancer and the big ones would be a lot easier to fix than they are. So I'm going to preface this whole presentation with that statement. I stole this from Miss Torres, and this is her presentation in its entirety. I haven't done much with it at all. Again, this is true. What I'm about to show you is true, but it is certainly not the most common. It does not take into account epigenetics. It does not take into account environment. It does not take into account transcription factors and RNAi and all the controls on expression that we've looked at so far. So here we go. This is basically for terms. First of all, if you look up Cleopatra's family tree, this is just an interesting intro. You'll see that it's really messed up. While historically she was apparently a very beautiful woman, uh, genetically she was rather mundane because she married, well, people in her family married their siblings and children and such. The problem with that is really simple. You have a gene pool, and that is the people that you can mate with. And if you think of all the people you can mate with as a bag of genes, or yeah, bag of genes, then um, if you have a uh, if you have an allele of a gene that causes disease, then if you marry someone from outside of your gene pool, the chance that you reach into that bag and pull out the exact same allele is rare. And actually, this is another reason that the genetics problems that you see a lot of times are misleading, because a lot of times you'll see only 2 alleles, and that's usually not the way it works. There's usually many more than 2 alleles. To say that evolution has produced 2 consistent mistakes over millions of years, I think is a little bit ridiculous. But the likelihood, if you marry into a gene pool that consists of your own family, then the likelihood that there's a copy of your disease gene in that gene bag is much higher. So when you marry into your own family, you basically don't save yourself. So if you have a disease allele, you're not saving or rescuing yourself by using somebody else's wild type allele. Okay, so these are some of the disorders that are caused by rather simple genetic problems. So we're going to take a look at a few of these here in a second. So this is the one of the problems that I abhor, but it's very simple. There is a color gene. It has 2 different alleles. We designate one big R and one little r. Again, that is not the way you'll see it in papers. So we've discussed how we'll see it in papers, and that's probably like OB++ or um, OB superscript 236, depending on the mutation or whatever. But you won't generally see big R, little r. But big R codes for the protein red. Big R makes a red protein. Little r makes a white protein. So if you have a big R and a little r, then you get a pink flower. If you have a little r and a little r, you have a white flower. And if you have a big R and a big R, then you have a red flower. You can see the Punnett they set up here. What they're basically trying to figure out is if you took this pink flower and you mated it with another pink flower, what are the chances that you would get certain alleles. And you should know how to do this. We would set this up differently. This would be 0.5 R, big R, plus 0.5 little r times 0.5 big R plus 0.5 little r, so that it would work out to 0.5 big R plus 0.5 little r squared, which would basically give you a quadratic, and percentages of 25, 50% for the pink and 25 for the white. This is, we call this incomplete dominance because neither gene or neither allele is really dominant over the other. Again, I don't even really like that term dominant, but when you have a third phenotype generated by the heterozygote, so the one that has one allele of each, you generate the phenotype of pink. So that means that you have a third phenotype that's called incomplete dominance because neither is dominant. Although both would be dominant, both of these alleles would be dominant over the R, super mini R, which would produce a clear protein or no protein at all. Yeah. All right, so this one is called codominance. And again, if you just understand the way these proteins function with each other, then it becomes really simple. So this gene is named I, and the different alleles are named IA, IB, and instead of little I, I'm going to say we're going to use I naught or I0. So this would be I0 and I0, and this is why I'm going to use these. So if this is a red blood cell, we talked about HLA markers in cancer. Those are the things that are presented to the T cells, but they're also the proteins that exist on the outside of cells that allow other cells to recognize them. So if you're thinking about a metaphor, it would be something like I think they're called ILS, ILS indicators in planes. So they're the little signal that planes send out in response to another signal that they get that say, look, we're the good guys or we're the bad guys. So I don't know if you know this, but radar often works on this very simple principle where you send a radar beam up to a plane and basically it sends you one back in response saying, I'm a good guy, I'm supposed to be here. If your plane doesn't send you that response, then you can, I guess you can assume that it's a bad one. But I0, I0, the genotype, produces no surface proteins. Now let's say we were our body and we're trying to destroy non-us cells, then we would destroy any red blood cell that had anything but this kind of clear outside shape. So we would destroy anything with carbohydrate A on the outside or anything with carbohydrate B on the outside. So if I took this blood cell and put it into us, and we have the I0, I0, then we would destroy it. If I took the B, this one, and put it in us with the carbohydrate B on the surface, we would destroy it. And if I took this one, a combination of A and B on the surface, and put it into us, we would destroy it. Okay, so if you have the alleles IA, IA, then you generate carbohydrate A on the outside. Actually, you anchor carbohydrate A with a protein on the inside, but whatever. I would then destroy carbohydrate B, but since there's no markers on I0, I0, I would not destroy that one. I would only destroy things with carbohydrate B. So then I would destroy this one with the circles on the outside, or this one with circles on the outside. But I would not destroy this. That's why if you are IA, or blood type A, then you can receive blood from blood type 0. And then you can see the rest of the pattern here, IB, IB, and IA, IB produces these phenotypes. It's a really simple idea. This is called codominant because if I have IB and I0, then I will have a cell that looks like this. So B is technically dominant over this. A is also technically dominant over this. A and B together are incompletely dominant, and if you consider the relationship between A, B, and 0, then A and B are codominant over 0. And again, this problem, these terms really become outdated fast when you're looking at real diseases and you have complex relationships and more than three phenotypes and more than, or sorry, more than four phenotypes and more than three alleles. This is what I was talking about the anti-A and anti-B. So you make antibodies if your blood type is 0 against A and B. If you are A, you make antibodies against B, and if you are B, you make antibodies against A, and if you are AB, you can't make antibodies against anything, which is sort of good because then you can get blood from anybody. And this isn't really true. So it turns out that there's a lot more markers on the cell surface than just A and B, and you know of them. For example, positive and negative are also blood cell markers. And this also generates the problem with tissue. So if you want to donate tissue, like a piece of a liver from you to a friend, you can't just donate it to somebody with the same blood type. There's a lot more matching that has to go on because of all the other things on the cell surface. I don't want to discuss that one. Oh, okay. This is kind of a cool one. This is sickle cell. So up here is the hemoglobin gene. It's on this locus or location on the chromosome 11, and there's two different versions. There is the normal version, and then there is the sickle version, which causes these long chains. So you can see that the hemoglobin is generally made of these four pieces, and here those four pieces form a line. Here the four pieces form kind of a squarish circular shape. So when they're in this squarish circular shape, they form nice healthy red blood cells that look like this. When they're in these long lines, they stretch out the blood cell, and when they go through vessels like this, they can get stuck or they can bust. And what happens there is that you just don't basically have enough functioning red blood cells, and you have a hard time getting oxygen from the air into your muscle so that you can break down glucose with oxidative phosphorylation. Okay, so that's not really a bad thing, though. Sickle cell disease actually has its advantages. But here's the explanation of kind of the problems it generates. But here's the crazy thing. So if you have a combination of the wild type hemoglobin allele and the sickle cell hemoglobin allele, you actually can generate, it can actually be good for you. So if you have, for example, a, what's the disease called? Not sleeping sickness. Brain. Malaria. There you go. Okay, so if you have malaria, malaria is actually, it gets itself into red blood cells and then it grows within the red blood cells. And then when it develops, it bursts out and destroys the red blood cell. So it turns out that if you have kind of a heterozygous, if you're heterozygous for this allele, so if you have the wild type hemoglobin allele and the sickle hemoglobin allele, then your cells actually don't stick around long enough for that parasite to develop. So in that condition, in that environment, the sickle cell is actually a bit of an advantage over the normal hemoglobin, because if you have the normal hemoglobin, then you have malaria all the time, and then you have bad dreams. You can look at this more if you want to. Sickle cell is not as simple as it sounds. It certainly causes some very, very problematic phenotypes, which you can see down here. It's also an example of a allele which is found predominantly in different concentrations in different populations. So African Americans have a much higher incidence of sickle cell than do Caucasian Americans. I really don't like that term. White people, European Americans, I don't know, not African Americans, I guess. I'm definitely gonna get in trouble for this. Oh boy. All right, so polygenes and epistasis. These are like the last two things that are normally mentioned in genetic chapters. And basically, polygenes means that there's many genes that influence one trait, and that epistasis is kind of the opposite when you have one gene and it modifies many other traits. And this is really the way it is for most alleles. This is kind of the model we've been studying. where you have many, many genes affecting a phenotype, and a single gene affecting many phenotypes. And then the fact that those genes have to interact with the environment as well to produce a phenotype, as we saw with the agouti. This is an ugly dihybrid cross, which you will never ever use in your entire life. But you can pretty easily use the way I showed you to use probabilities to generate this kind of a table. Again, you can use Pascal's triangle. This is the Punnett square. It's a big one, but you can use anything to expand this quaternomial. These are puppies. I like puppies. And that's the end. No. Seriously about the puppies, well, more seriously. This is kind of when you're having two different alleles, or two different genes, sorry, not just alleles, two different genes affect coat color. And so this is a case of your hair too, where black and blonde and brunette are actually combinations of two sets of different colors. So I think you have a black gene, so there's like a black version of the gene and a brown version of the gene, and then there's the E version, which is, I think it's like reddish. So let me see if I get this right. It's so for example, this guy is black and or brown and red. This guy is just brown, and this guy is not red and not brown or blonde. I'm pretty sure I've messed that up. And if this affects your future science career, I apologize. If ever you are in a situation where you're in a lab and it comes down to a life and death situation where you need to predict the phenotype of a dog by looking at the genes B and E, and I apologize, but as it is a life and death situation, it probably won't matter anymore because someone would be dead. Anyway, have a good day. Bye.